in cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide in cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide. An interphase, the cell gets big. It makes a copy of DNA. The cell is active, matures in size, prepares to split into two sides.
I know the next stage is called mitosis. It's prophase, metaphase, that's not all. And three anaphase, and the next telophase. Then inside of phonesis, the cycle is done.
Prophase is one, the nucleus. Chromatid condenses, making chromosomes. Spindle fibers form a bridge between end of the cell, and then the membrane breaks down. Let's do it, let's do it, let's do it, let's do it, and do it, and do it.
Let's live it up
and do it, and do it, and do it, do it, do it. Let's do it, let's do it, let's do it. Come in cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide in cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide. Metaphase is next. Hey, chromosomes line up. All in the middle. Hey, and they hold up. The spindle fibers at its centromere, which still is holding. Come on, chromatids joy. Anaphase is up. 3. It's time to split away. Centromeres are breaking. They separate the chromatids. It moves along. The spindle fibers, opposite ends of the cell. Let's do it, let's do it, let's do it, let's do it, and do it, and do it. Let's leave it and do it, and do it, and do it, do it, do it, do it. Let's do it, let's do it, let's do it, do it, do it. Telophase chromosomes begin to stretch out. They don't look like they did, no more like rocks. This occurs at the end of the new cell. Nuclear membranes form around these parts.
mitosis, interphase, prophase, metaphase, anaphase, telophase, cytokinesis.
Do it!
Cell membranes go around the middle of the cell. Cell branches into two at the daughter cells. The
Cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide in cell division. Events make cells grow and divide. Events make cells grow and divide. Events make cells grow and divide. Cell division.
Hey there, boys and girls. Welcome to our vodcast on cell division and mitosis. What we're going to talk about is the function and the purposes of cell division in our bodies, and we'll talk about the process of the cell cycle and what mitosis does and the stages of it. Let's take a look and let's get started. Okay, in the back of your foldable, you should have 2 sections: why is cell division important, and the 3 results of mitosis. So you should be looking at the back of your foldable right now, and we're going to be filling out why cell division is important. One reason why cell division is important is it pertains to single-celled organisms. Since single-celled organisms are only one cell, they don't make other cells to help reproduce. So in order for single cells to reproduce, they basically split in half. So as we talked about with bacteria undergo a process called binary fission, where the bacteria grows, duplicates its DNA, and splits in half, as we see here on the left. So when we talk about cell division, ladies and gentlemen, we're not exactly reducing the amount. When cell division occurs, we're actually making more. So in this case, division is multiplication. So single-celled organisms reproduce using cell division. Now, when we talk about multicellular organisms like you and I, we're made up of trillions of cells. Cell division has a couple different functions because we don't need cell division to reproduce. So one of the reasons why we carry out cell division is because multicellular organisms use cell division for repair. If you take a look at this diagram here on the left, this is an x-ray of a broken forearm. So if you're not sure where that break is in the forearm, here it is. And you see where the bones don't match up anymore. So when you break your forearm, you go to the doctor and they put it into a cast. And after they put it into a cast, you can't use that arm and move that arm for a while, because they're setting the arm. And the reason why they're setting the arm is to keep the bones in position, because what's going to happen is that your body is going to produce more bone cells to repair that break and reconnect the bones into a healthy forearm. So that's one way your body repairs it. If you tear muscle tissue, your body makes more cells to repair that muscle tissue. This is one example why cell division is important in multicellular organisms, because we use it to repair tissues. However, there is another reason why cell division is important in multicellular organisms. Now, as we talked about earlier this year, when multicellular organisms grow, their cells don't get bigger, they just make more cells. So if we take a look at the sequence of events here, if we start at the upper left-hand corner, here we have one cell, a fertilized egg, that divides into two cells. And these two cells divide into 4 cells. And then the process continues until we get into what's called a hollow ball of cells. And then this hollow ball of cells will become an embryo. And this is what we all look like at one point in time in our lives. And then that embryo, as it continues to divide and produce more cells, is going to start to take shape of a human baby. So this is actually a photograph of my daughter, Riley, at 30 weeks or something like that. As the cells continue to divide and develop inside the baby, when the baby's born, they're going to look something like this. So this is my daughter at 10 days old. And because she's a baby, just like you were at one point in time, your body's going to continue to make more cells, and you will get bigger. As the year passes, this is my daughter at 1 year old. So as you can see, there's a stark difference between what she looked like at 10 days and what she looked like at 1 year, is because she was growing. The reason why she was growing and continues to grow today is because her body is continually making more cells each and every day. So multicellular organisms use cell division for growth and repair of tissue. Now, how does this all happen? Well, it all happens in a process called the cell cycle. So let's take a look at the cell cycle. All right, boys and girls, this is a chart that shows you the 3 parts of the cell cycle and how much time is spent in each parts of the cell cycle. So here we have this big blue section of this chart here. And this section here represents the phase called interphase. So this is the first part of the cell cycle. Now, interphase takes up a lot of time. That's why this chart here is mostly blue. And once we get out of interphase, we then enter a process called mitosis. So that's the second part of the cell cycle. And after mitosis is finished, we enter the last phase called cytokinesis, where the cytoplasm divides. So what we're going to talk about here is we're going to now take a look at an in-depth look at what exactly happens in each one of these phases. So you might want to open up your foldables to the insides now and get ready to take your notes. For this section of the notes, boys and girls, you're going to have to listen to the narration in the video to fill out your questions. So make sure that you read what questions it is that you need to answer, and then fill them in as we go along. So make sure you stop and rewind and do all the things you need to do in order to get the right answers. So let's start taking a look at our different phases of the cell cycle. So as we said, the first part of the cell cycle is called interphase. And this is the part of the cell cycle where the cell grows, and then the DNA in the nucleus begins to duplicate. The DNA duplicates because we're going to make a second cell, so we have to make another copy of DNA for that. So as we learned in the last chapter with DNA replication, the DNA is going to split and make 2 identical strands. Now, as we get out of interphase, I just want to show you, so this makes a little more sense. This cell has 4 chromosomes in it. We have a green chromosome, a blue chromosome, a red chromosome, and a pink chromosome. So once we're out of interphase, we then move into a phase called prophase. I always remember prophase as the preparation phase, because now things are going to start to happen to get ready for the nuclear division. Before we move on, we need to learn the difference between what a chromosome is and a chromatid pair. In your cells, you have chromosomes, which are just DNA wrapped up around proteins. Here we have a regular chromosome. It hasn't undergone replication yet, so it hasn't made a copy. However, when it does make a copy, as it will in interphase, you'll notice that it becomes like this X-type structure. So you can think of it as one half of the structure is the original chromosome, and the other half of the structure is the copy of that chromosome. And when we get the both copies of the chromosomes together, you get what are called chromatid pairs. So the chromatid pairs are always going to be referring to this X-shaped structure you see. Now in the middle here, you'll notice that the chromatids are connected at a point called the centromere. I always remember the centromere is where they connect because it's in the center of the chromosomes. So when I refer to chromatid pairs, I'm referring to the X-shaped structures here. And when I say chromosomes, I'm referring to the actual individual chromosome by itself. Alright, so as I said, prophase is the preparation phase. So these are the things that are going to happen to start prepping the cell for this nuclear division. First of all, the DNA is going to condense, forming the chromatid pairs. Now here in interphase, you don't see the DNA replicating itself, but once it does, they start to condense, as I said, and become more visible in the prophase part of mitosis. So this is where you can actually see the chromosomes, and this is where the X shapes come out. So as we said, here's your chromatid pairs. The next thing that happens, as the chromatid pairs are condensing and showing themselves, is that the nucleus begins to disappear. In addition to that, these structures here called centrioles are going to start to move to opposite sides of the cell to carry out the cell division process. So prophase is to get everything started to get the cell division going. Once we get out of prophase, everything else is pretty simple and straightforward after that. We move into the phase called metaphase. And metaphase is known as the middle phase. And the reason why I call it the middle phase is because the chromatid pairs line up across the middle of the cell. So as you can see, here we have our chromatid pairs right down the middle. So middle meta. That's how I always remembered it. What you'll also notice is that the centrioles, when they move to the opposite ends of the cells, they also form these fibers called spindle fibers. So these spindle fibers move across and attach to the chromatid pairs at a point called the centromeres. So now our chromatid pairs are attached to the centrioles by these spindle fibers at the centromeres. Once that happens, we move on to the next phase called anaphase. I always remember as the apart phase, because in anaphase what will happen is the spindle fibers will shorten and they pull the chromatid pairs apart at the centromeres. So they kind of look like wishbones. If you pull on a wishbone with a friend, both halves are going to go in opposite directions because it's being pulled apart. So the apart phase is anaphase, as the chromatid pairs are now being pulled apart from one another. And after anaphase is done, we move into telophase. And I remember telophase as the two nuclei phase, okay? Because two nuclei is what's formed. So here's one nucleus, and here's another nucleus. The second thing that happens is that the cytoplasm of the cell begins to pinch. So you have the splitting cytoplasm here starting to pinch off and then divide into two cells. And in addition to that, you'll notice that the spindle fibers are now gone. Those are the things that are happening in telophase, and once that all happens, then we get into the phase called cytokinesis, the third and final part of the cell cycle. And in cytokinesis, the cytoplasm completely pinches, separating the two cells, and then out the cytoplasm divides, forming two genetically identical cells. So those are the stages of the cell cycle: interphase, mitosis, and then the cytokinesis. Now there are a couple things that you need to remember about mitosis. So you want to flip to the back of your foldable right now and take a look at the 3 results of mitosis and write these down. Mitosis is only the division of the nucleus. It's not cell division, it's the production of one nucleus into 2. The 2 nuclei that are produced are identical to one another and to the original nucleus. So they're identically the same in terms of their genes. They have the same exact genetic material. There is no difference between them. And then 3, each nucleus has the same number of chromosomes and same types of chromosomes. So those are the 3 main results of mitosis. Now let's take a look at an animation here to put it all in motion and hopefully give you a better idea of how this all works.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis, or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Okay, I hope the animation was good. I hope that cleared up things if you were a little confused. And what I want to do now is just kind of show you how the chromosome numbers change and then come back to being the same. So if we take a look here in the diagram up at the top right, this is interphase, and as we said, the chromosomes are going to replicate. But I just want you to know again, this cell is a 4 chromosome cell. We have 1, 2, 3, 4 chromosomes. When in the beginning of interphase, this cell is going to have 4 chromosomes. And as we said before, during interphase, the DNA is going to replicate. It's going to copy itself. So as a result, we're going to go from 4 chromosomes and double that number to 8 chromosomes. When we get into prophase, that's when we see our chromatid pairs again. And if you're wondering where the 8 chromosomes are, all you have to do is count. 1, 2, 3, 4, 5, 6, 7, 8. So prophase, we have 8 chromosomes. And then in metaphase, as they line up, you can easily see the 8 chromosomes still. So we still have 8. And then in anaphase, you can see 2 sets of 4 chromosomes being pulled apart. So as these chromatids are being pulled apart, we still have a total of 8 chromosomes in the cell. Once the cell divides and the chromosomes are pulled to the opposite sides and the nucleus forms, those chromosomes have now been separated from one another. So one nucleus is going to get half the genes or half the chromosomes, so it's going to have a total of 4 chromosomes. And then the second nucleus is going to have a total of 4 chromosomes. So if you take a look here, you'll notice that the 4 chromosomes are moving to this side, and the 4 chromosomes are moving to that side, leaving this nucleus with 4 chromosomes and that nucleus with 4 chromosomes. And as a result, as the cell divides and pinches the cytoplasm, you're going to have a cell with 4 chromosomes and a cell with 4 chromosomes. And the final number here is the same exact number we had to start with. So that's how the chromosome count works as we talk about mitosis. Now, what does that mean in humans? Well, if we take a look at humans, we all have 46 chromosomes inside of our body. So I have 46 chromosomes, you have 46 chromosomes, your parents have 46 chromosomes, your friends and relatives have 46 chromosomes, and our main man here, Batman's got 46 chromosomes. So what we're going to do is we're going to start out with 46 chromosomes. And then again, in interphase, we're copying our DNA, so we're going to be copying our chromosomes to give us a total of 92 chromosomes. Because again, we want to put another set in the new cells. So as a result, as we move into prophase, we're still going to have 92 chromosomes, and then those 92 chromosomes are going to line up in the middle during metaphase, and then those 92 chromosomes will get pulled apart. Now in our picture we have 4 going to the sides, but again we're working with 92. So that means 46 chromosomes are going here, and 46 chromosomes are going here. And as a result, since 46 chromosomes are being pulled to one side, and 46 are being pulled to the other, the new nucleus here is going to have 46 chromosomes, and the second new nucleus is going to have 46 chromosomes, which means the cells are going to have 46 chromosomes. Now, one thing to remember about this is how your body cells make new cells. Mitosis produces every cell in your body except for sex cells. Sperm cells and egg cells are not produced by mitosis. They're produced by a different cell division cycle. However, you fall down and scratch your arm, the new skin that forms underneath the scab, those cells are made by mitosis. You cut your hair, and your hair grows longer within the next couple of weeks, well, that's mitosis creating new hair cells to lengthen your hair. You break a bone in your arm and the doctor sets it, the new cells that patch up that break, those cells are made by mitosis. So all of your body cells are produced by mitosis. So that concludes tonight's vodcast, boys and girls. Thank you for your time.